Hello, everyone. I'm Oluwadamilola M from the University of Georgia. I want to present our innovative research on quantitative and biological evidence for health-dependent space use dynamics in broilers. There is an increasing trend in broiler production around the world, and statistics has shown that 14 to 30% of the broilers being produced experience mobility impairment. It affects productivity and welfare. It causes pain, discomfort, and restricts movement. Due to the restriction of movement, it reshapes how these broilers interact with their environment. And currently with technologies, we measure mobility impairment indicators such as movement features that will measure things like the steps, the walking distance, the strides. We also measure gait score. Gait score involves evaluation of the working ability of individual broiler chicken and assigning a score to them based on a standard scale. Then post estimation measures or examine the key points of the chicken body area and check for changes or postural defects. There's something common to all these indicators. They are episodic, they are non-continuous, and they cannot tell us how this mobility impairment affects the animal itself or how it affects the animal environment interaction. It is worthy of note that mobility impairment is not only a locomotory postural problem, it is also a spatial behavioral problem because it alters how these broilers use space and resources over time. These are the reasons why we need to move from episodic evaluation to animal environment interaction based using spatial behavioral matrices. With spatial behavior, the indicators we need to measure are the space use diversity, the movement, decisions, and preference of these broilers, and how they changes over time. And that is the space use dynamics. We formulated an hypothesis, and the hypothesis is that broiler leg health significantly influenced broiler spatial behavior. If the hypothesis is true, spatial behavioral metrics may be useful for evaluating and predicting broiler leg health problems using computer vision. The study objective is to quantify and compare the spatial behavior of broilers with healthy and unhealthy gait at week 4 to 7 of age. Now, I'd like to go to the materials and methods. This experiment was carried out at the University of Georgia Poultry and Research Farm. 1,776 day-old COP 500 broilers were allotted to 48 pen space. Each pen contains 37 broilers, and out of the 37 broilers, 10 of them were color marked for identification and continuous monitoring. This is a workflow. At weeks 4, 5, 6, and 7, at the end of the week, we gate scored the broilers on a scale of 3. Any broiler scoring 0 is classified as healthy, and the broiler scoring 1 to 2 were classified as unhealthy. Thereafter, we move on to video record these broilers continuously in the pen space. And then we developed a tracking algorithm to track the broilers individually and extract their spatial location coordinates. Then we move on to track them based on the zones within the pen, and we carried out a spatial behavioral analysis. This video shows the tracking process. We assign an identity to each of the individual column marked chicken. We extract the X and Y coordinates of the bonding boxes. Then we move on to quantify the spatial behavior. To do that, the first thing we did was to divide the pen space into 4 zones. The first zone, first one is a feeding area. The second one is a drinking area. And the third one is the corners, and every other space in the pen was categorized as open spaces. Now, we associated the X and Y coordinates we obtained earlier from the tracking to these spatial zones. It means that at every point in time, each of the broiler belongs to one spatial zone or the other. And as they move from one spatial zone to another, the trajectories enable us to do spatial behavioral analysis. The indicators we measured is a space use diversity, in terms of the Sharon Diversity Index. We checked for the species diversity between healthy and unhealthy birds, and we checked for the drivers of these differences. We also analyzed the spatial compositional structure, and we checked if this broiler showed preference for resource area or not. Now, I'd like to go to the key results and interpretation. These are the graphs explaining the Shannon Diversity Index. We could see this brown bar represents the unhealthy birds and the blue bars represent the healthy birds. We could see that the unhealthy birds continually showed a higher Shannon Diversity Index than the healthy birds. This indicates that the unhealthy birds exhibited significantly higher species diversity than the healthy ones. And we can see that this species diversity decreased with age in both groups. And the largest differences occurs at week 4 and week 5. Now, we move on to check for the drivers of these differences. When we checked for the driver of these differences, this panel, this area explains the main and the major driver. This is the second most important driver, this is the third, and this is the least most important driver. So we could see now from the graph that the most important dominant driver at week 4 to 5 is the drinking area. This is for week 4, this is for week 5. And we went ahead to check who are the key users, and we found out that the key users are the unhealthy birds as implied by the negative mark in this. Then, in these compositional differences structure, we saw that there is a trade-off between the use of drinking area and open space or feeding area. This implies that the unhealthy birds that intensely use the drinking area, they did this at the expense of the feeding area and the open space. Now, based on this intense use of the drinking area, we went ahead and checked if this bird shows a significant preference for resource area. And yes, leg status influence feeding and drinking area preference. This first graph represents the drinking preference of the birds. And we could see from this graph that the brown bars represent the unhealthy birds. They showed significantly high preference for the drinking area compared to the healthy birds. And we could see that this preference for drinking area increased and peaked at week 6 before a decline. But the reverse was the case for the feeding area preference. Now, this result indicates that spatial behavioral changes involves resource use allocation. We move on to week 6 and 7 to check the dominant driver of the diversity differences we saw earlier. And interestingly, there is a shift. Now at week 6 and week 7, the corner became the dominant driver of these differences. And there is a trade-off between the usage of corner area and the feeding area and open space. We check which birds are the key users, and we discover that the unhealthy birds are the key users of this dominant area. It means that the unhealthy birds used the corner area more intensely at the expense of the feeding area and open space. Now, the biological interpretation for this. We can see from this video that the unhealthy birds leaves a red line. So these unhealthy birds move from the corner area to the drinking area to the corner or feeding area. They always pass through the open space. And this is the reason why these birds exhibited significantly higher space use diversity, because they utilize all the zones in the pen. Unlike healthy birds concentrated their activity just in the feeding area and the open space. They also go to the drinking area without going into corners. Now, the higher space use diversity exhibited by unhealthy birds could be an indicator of constrained time redistribution across pen space. At first, these unhealthy birds exhibited resource-seeking behavior, reflecting selective movement decision around the drinking area, and this is primarily for energy conservation. Then, with the worsened leg health, they showed dominant corner-seeking behavior in area with low effort and less disturbance. The welfare implication of this is that in the traditional leg health detection, we measure locomotion and locomotory posture. But with spatial behavioral analytics, we measure the behavioral consequences of this leg health. And the application is in welfare analytics, automated welfare monitoring, and farm management decision support. The key take home from this: number one, higher species diversity does not necessarily indicate better welfare. Secondly, unhealthy birds may show more even but behaviorally constrained species. And unhealthy birds transition from resource-driven pattern to corner-based spatial segregation. Mobility impairment from week 4 to 5 modifies movement decision, but at week 6 to 7, it restricts spatial freedom. And those spatial behavioral matrices could be useful indicators for distinguishing birds with healthy leg and unhealthy leg through computer vision tracking. I want to acknowledge our collaborators, the founders of this project, my lab mate, and my advisor, Dr. Gumili. Thank you for your attention.